Well, so for my study, we're going to be looking at exposure to butyl benzyl phthalate during pregnancy and lactation and how this alters maternal mammary morphology, as well as progesterone receptor expression in our Balb mice. So environmental contaminants are ubiquitous and pervasive in our environment. Now, many of these chemicals have shown to have significant effects on offspring mammary gland development when exposed to the pregnant mother. Now, this is done either by mimicking or disrupting normal endocrine signaling. Much effort has been made to explore the effects of maternal exposure on offspring development, but what about the mother herself? The adult mammary gland is dynamic, undergoing growth, differentiation, and regression from the onset of puberty all the way to return to ovulation. So essentially, the mammary gland itself is going through an adult form of development. So, could the same exposure that disrupts offspring mammary gland development also disrupt maternal mammary gland development? So to study this, we exposed Balb mice over the course of pregnancy and lactation to butyl benzyl phthalate. So this is a chemical that is commonly used in PVC pipes, as well as some other plastic components as a plasticizer. It comes into contact and therefore can contaminate water. We used 3 different doses of butyl benzyl phthalate. These were calculated to provide a range of concentrations that were relevant to humans. In fact, these doses span from actual concentrations measured in humans to those that have been assessed and determined as non-toxic. Now, after weaning, mothers were left until 5 weeks after involution, or this is the regression step that brings us and returns the mammary back to a mostly pre-pregnant state. Now, all treatments then were compared back to both pregnant and non-pregnant controls, which received only a corn oil solvent. For our results, we first evaluated whole mount maternal mammary glands for morphological differences. This figure here shows the percentage of the mammary composed of ductal tissue, and we can see that there's a significant increase in the ductal epithelium in the 3 microgram dose compared to that of the actual pregnant control. Next, we evaluated progesterone receptor. So progesterone receptor is a key driver of ductal branching and expansion of ductal tissue. We can see here in this figure that the percent of cells positive for progesterone receptor are significantly reduced in the 500 microgram dose compared to those in the pregnant control. Now, this is pretty interesting considering the fact that we just saw in the previous figure that there was an increase in this ductal complexity. In addition, but not shown here, we also evaluated a proliferative marker to see if ductal increase was actually due to increased cell proliferation. And also interesting is we saw no differences between these groups. So what did we find altogether with this study? Well, first, butyl benzyl phthalate exposure during pregnancy and lactation does in fact alter the maternal mammary gland. It does this by increasing ductal tissue while decreasing progesterone receptor and seems to not have at least a noticeable effect by us on proliferation. Furthermore, and quite important, is the fact that these changes are occurring at concentrations that are relevant and directly applicable to human exposure. Last, we can see then that we do need to have future studies that focus not just on offspring developmental alterations, but those as well on the mothers that bear them. And I will be happy to take any questions from them.
Great, thank you so much. Why don't I kick it off with a first question?
Sure.
My question is, can you speak a little bit about the dose response? It didn't look like there was a very clear dose response, particularly in your first slide, which I think was the ductal volume. It looked like there was a difference, between the pregnant control and the 3, but then it wasn't maintained to the higher dose of the, 500 and 18,000.
Yeah, absolutely. So as we can see across this, all the means are slightly increased, but the only significant difference we actually find is with the low dose. Now, that's not totally unheard of. In fact, this is something that's been reported quite a bit from Laura Vandenberg, my advisor, is that you can actually have really powerful effects that come at low doses specifically. And now I can't say for sure, but I can suggest that potentially any kind of disruption to the normal amount of steroid signaling that can occur can also then have response and feedback mechanisms. And so oftentimes you can have very powerful effects that can kind of sit underneath the response of that feedback.
Great, and then I'm gonna go a little bit out of order in the questions that came in because there's a question that's related to the dose response question from John Serrard asking why the U-shaped curve for the progesterone receptor?
Okay, so again, now another type of thing that we can see, of course, are non-monotonic dose responses. which again can be related to these feedback responses. Now, we don't actually have a significant difference between the 3, the 500, and the 1800 altogether, right? These aren't all completely different. So I can't actually say that there is indeed a, for sure, non-monotonic dose response. That is something that would be interesting to evaluate. And again, it may be possibly explained by some of these negative feedback mechanisms that also guide progesterone signaling.
Great. And there's a question from the Dean on how would you conduct this research in humans?
Oh, that would be an interesting one. So for certain ethical considerations, I wouldn't certainly be dosing humans. I suppose this would be something that might be best looking in epidemiological study, because this is really going to be dependent on exposing humans directly to the treatment itself. So I guess I'm not quite adept at answering that. I think for direct human experimentation or epidemiological evaluation.
Great, thank you. From Monica Roy, who said you did a great job.
Thanks, Monica.
Of the concentrations you tested, which ones have been measured in humans versus in the environment?
Okay, so the highest dose is, so it's the 18,000 micrograms per kilogram per day. So that was actually from the no observed adverse effect limit. So this was something that was calculated within the actual models. But when we look at the 3 microgram per kilogram per day, so this was actually taken from studies that looked at the 95th percentile exposure for pregnant women. So this was something that was actually found. It's the high end of the pregnancy exposure, but this is something that was relevant to humans and taken from human study.
Great. Any thoughts as to how the mammary ducts can increase in volume while decreasing in progesterone receptor and not changing in rates of proliferation?
Sure. So in our study, we evaluated these animals post-involution. So this means that it's actually post-regression of that buildup that we had in pregnancy and lactation. So we're kind of getting a snapshot somewhat after the fact of this process. Now with this, if we were looking at animals during pregnancy, we might expect there to be a ton of proliferation. But at this point, we actually may see what is the result of changes to the lack of regression in the processes that drive that. So as an example, there are 2 parts that occur in involution primarily. So one is going to be a gene expression change that will trigger a significant amount of apoptosis within the mammary ducts. This is then followed by an immune response. So one thing that we'll be evaluating here, once we could get back in and hopefully when this COVID thing ends, we can actually take a look and see, well, do we see increases then in immune response or increases in apoptosis that can explain how we can have more ductal glands in these exposed animals as opposed to, without having that proliferation.
Great, and I think I'm not seeing any other questions. I think I'll ask one last question that I had, and that was, is there anything known regarding these exposures on the pups? You mentioned early on that is where there tends to be even more research than on the, the mothers. So
yeah,
what is known about the
So butyl benzyl phthalate itself has been somewhat poorly studied in general. So I treat this a little bit somewhat as an example that contaminants can expose the mother itself. Now, there are a number of other chemicals that have been studied at a much higher rate. So things like, for instance, bisphenol A has been a very popular study mechanism as an estrogenic chemical. But something like this, and as an example that butyl benzyl phthalate can represent, is the fact that it's something that happens to the pups, or a model for something that happens to the pups could actually happen and have changes within the adult as well. But we actually do have some pups that will be coming from this experiment. I don't have finalized results on those available, at least to me right now. So hopefully I will actually be able to give you some information specific to what is going to happen in these pups that were exposed through these mothers.
Great. I think we're all set. Thank you so much for a great talk.